Hello, everyone. Good morning, good afternoon, good evening. Please feel free as we get settled and are ready to begin our session. You can tell us where you are logging in from in the chat. We always like to see who is logging in and from where. So I'm from Washington, D.C., USA. Glad to see you all. Freiburg. Good afternoon, Freiburg. All right. And I believe as you all introduce yourselves, and thank you for doing that. This is a wonderful community. We will get started, and I'd like to welcome you all to today's distinguished lecture. I am IBS Executive Director Peter Doherty, and I'd like to thank you all for joining us today. Since its beginning in 2022, the primary purpose of this series has been to encourage young professionals globally to pursue all the possibilities available to them through this profession by offering inspiring talks from accomplished lecturers in the field. And before I introduce today's guest presenter, we are privileged to have with us as moderator International Biometric Society Education Committee Chair Rafael de Andrade Maral, who will help facilitate the Q&A portion of today's session. We appreciate the Education Committee's diligent work in identifying exceptional speakers for this series. That's one of their charges, and for offering ongoing support to this project. And Rafael, thanks very much for joining us. We will hear from you a bit later. I'd like to take just a moment to mention that we have a very special event coming up next year. It's IBC 2026 to be held in mid-July in Seoul, Korea. This is an opportunity for everyone in our community to take part, either as an attendee or as a speaker. And we are in the midst of our abstract submission period for contributed sessions. We've also just completed the abstract submission period for several other groups of sessions, our invited sessions, and also our short courses, and we will have more news on that soon. But please make plans to join us in Seoul. We have some very innovative tweaks to our program that we hope you will enjoy. We've also tried to shave off about a day's worth of programming to make travel to and from Seoul a little bit easier for you. So we hope to see you then. Now on to today's session. We encourage participants to post their questions using the Q&A feature in Zoom. We're also happy to answer any additional questions following the conclusion of the presentation in the IBS member community. And just a reminder, in case you didn't know, that the community and the previous recordings in this series are available to all members free of charge. Xu Yang is professor of statistics at North Carolina State University. She received her PhD in applied mathematics and statistics from Iowa State University and postdoctoral training at Harvard T.H. Chan School of Public Health. Her primary research interests are causal inference and data integration, particularly in the context of comparative effectiveness research in health studies. And she also works extensively on methods for missing data and spatial statistics. She has been the principal investigator for several US NSF, NIH, and FDA research projects. And now, without any further delay, I'd like to hand the program over to Dr. Xu Yang. You have the floor, and thanks again for being here today.
Thank you, Peter. Let me share the slides. Can you see the slides okay?
Yep. Yes.
Okay, sounds good. Good morning, everyone. Thanks for having me. It is my great pleasure to be here for the IBS Distinguished Lecture. And today I'm gonna talk about the integrative approaches to treatment evaluation, how we can bring together randomized clinical trials, real-world data, and modern statistical methods to improve how we can generate evidence for healthcare. So the focus today will be on the interface of RCTs, RWE, and also causal inference, and how we combine these perspectives to help us address some scientific questions or challenges. And the research is supported by the FDA UN grant, and the content are our own and not necessarily represent the FDA's viewpoint. And here is the roadmap for today's talk. So I will start with the policy landscape and how it has acted as the catalyst for the use of real data and evidence. Then I will move to the interface of RCT, RWD, and the causal inference, machine learning, focusing on how we can combine conventional approaches with the new technologies. Next, I will introduce hybrid control trials as an example and a bias-aware frequentist toolkit for integrating external controls into the RCT analysis. Finally, I will wrap up with a summary and some thoughts on the path forward. So the research actually is motivated by the 21st Century Cures Act passed in 2016. It emphasizes two things. One is big real world data, and the other one is precision medicine. It is designed to help accelerate medical product development and bring new innovations to patients faster. And in response, the US FDA has developed a framework called the RWE framework that try to evaluate the potential use of real world data and real world evidence for drug and device approval. What is real world data and real world evidence? According to the US FDA, any data relating to patient health status or the delivery of healthcare is called the real world data. And it can come from a variety of sources, for example, disease registries, natural history studies, electronic health records, claims, so on and so forth. And any clinical evidence derived by analyzing the real world data is called the real world evidence. And we know that different studies have different designs and they can produce different levels of evidence for medicine. So here you can see the pyramid of evidence-based medicine. We know that randomized clinical trial has been regarded as a gold standard because we have a controlled setting. We can control randomization of the treatment, and therefore they can produce the highest level of research evidence. So you can see such studies usually are placed on the top of the pyramid. On the other hand, the real world data, because they are not collected for research purposes, so they suffer from possible biases. Therefore, such studies usually reside at the bottom of the pyramid. And this slide shows the growing range of clinical research designs from traditional RCTs to fully observational studies. Throughout this spectrum, we're seeing an increasing common use of RWD to support or even replace part of the traditional trials. So for example, in conventional RCTs, RWD helps to evaluate trial feasibility and enrollment by examining claims or EHR data to identify eligible patients or eligible participants. Then there are various study design that can combine RWD, such as programmatic trials, externally controlled trials, hybrid designs, and a target trial emulations from observational studies. So the overarching message is very clear. We are moving from a world where RCT alone were the gold standard to a continuum of the study designs that integrate RWD at varying level. And this slide shows concrete examples where the FDA has already approved drugs and biologics with real-world evidence included in the submissions since 2019. It really demonstrates that RWE, the real-world evidence, is not just a theoretical, right? It is already influencing regulatory approvals. So this signals regulatory readiness. And there are some criticisms about the real world data, but the US FDA is now open to using real world data and the real world evidence for drug and device approval. So the agency really believes that such data sources can and should be included if design and analysis is done appropriately. So on the left-hand side, you can see the guidance for industry produced from US FDA about how to utilize the real data for drug and device approval. And the global picture actually is similar. So on the left-hand side, you can see some guidance and considerations from the US FDA. In the middle, these are the initiatives from EMA, that stands for European Medical Agency. And on the right-hand side, you can also see some pilot programs from China and Japan. So this really shows this area is very active and evolving. And we also have very active discussion around real evidence in our community, including the ASA BioP section. So the ASA BioP section charted the RWE Scientific Working Group in 2018 to encourage statistical participation and also contributions to this active and evolving area. So the working group aims to understand and also advance statistical methods in a pre-competitive space and to promote the appropriate use of real-world evidence in support of medical product development and regulatory decision-making. And this year is phase 4, and the theme is around the hybrid designs, AI, ML, and the negative controls for generating reliable real-world evidence to support regulatory decision-making. We are encountering some challenges from emerging techniques like real data, real evidence, AI, ML, but we are also taking this opportunity they offer. So now I will move to the interface of RCTs, RWD, and also causal inference, AI, ML, focusing on how we can combine conventional wisdom with the new techniques. So let's understand RWD or RWE a little bit better by contrasting the complementary features of conventional RCTs and the real-world data. We know that RCTs are the gold standard, while RWD is an emerging evidence source that can extend or complement what trials can tell us. So firstly, RCT remain the gold standard because randomization ensures internal validity and minimize biases, right? However, they are often limited in representativeness. So for example, only 8%, about 8% of cancer patients actually participate in oncology trials, and they tend to have smaller sample sizes, short follow-up windows, and high cost. On the other hand, real data captures patients in routine practice, so making it far more representative of the populations who will actually use the treatment. So it allows inclusion of diverse patient subgroups, for example, older adult, minority, those with comorbidities, so on and so forth, who are often excluded from the trials. And also RWD offer larger sample sizes, longer follow-up windows, and a lower cost compared to RCTs. But unlike RCTs, RWD lack randomization, right? We don't have randomization of the treatment in the real world settings. So there is always the risk of unmeasured confounding. This is a challenging case for the causal inference. We want to make sure that causal inference methods can be applied to make the result credible. But from the table, it is very clear, rather than competing, RCTs and RWD should be viewed as complementary evidence source. And the future is about integration, not replacement. And we can actually achieve a lot by integrating RCT and RWD. The key objectives of the integration of RCT and RWD include the following. First, we can improve the generalizability of RCT findings so they can apply to broader, more diverse patient population. Secondly, to increase the efficiency and the power of estimation by leveraging external data sources. Third, we can enable long-term safety and effectiveness monitoring, something that RCT alone usually cannot achieve because RCT usually have very short window as a follow-up, and many others. But we need to be cautious, right? So the FDA has highlighted several concerns when using external or real-world data. First, the selection bias. That means that patients in the real-world databases may not be comparable to those in the trials. Secondly, unmeasured confounding. Key disease variables may be missing or inconsistently captured. Third, issues with timing. Real data may not align well with the trial period, right? And finally, but not least, outcome definitions may differ, and these are not minor details. They can fundamentally affect the validity of the conclusion. So while RWDs are promising, we must rigorously address these sources of bias. And the FDA's position is very clear. So innovation must not come at the cost of validity. So validity comes first. Now we need a clear roadmap. We call this clear causal roadmap when we're talking about integrating randomized clinical trials and the real data and combining statistics and AI ML, bring like the predictive power of AI ML into this area. So we begin with the causal assumptions, the causal questions, sorry, the estimate of interest that defines the question of interest and also the target population. And then we can clarify assumptions, articulate all the assumptions required, linking the causal estimate to some statistical parameters, and finally estimate them with the right tool. And the roadmap helps us clarify goals, identify the correct biases, and also articulate assumptions. Here in the estimation procedure, we actually can leverage like the predictive power of AI ML, right? Currently, the community have seen a lot of use cases of AI and ML. So AI ML here can help us model complex patterns and high dimensional relationships. So in recent years, we actually seen that AI ML can be embedded in causal inference, such as the W robust estimator, to model complex relationships between different variables. And at the same time, we can preserve some statistical properties, statistical rigors in the treatment effect evaluation. So this is where the causal framework can be very helpful to bring or leverage the predictive power of AI and ML. And data integration actually has been a very interesting research agenda. We have been developing causal inference methods for combining RCT and RWD. Here is the snapshot of the methodology we are developing. So you can see we have some summaries or review papers on this area. And also we have a GitHub repo. So if you are interested in implementing any of the statistical methods here, you can check out this GitHub repo. And here I divided the research into two lines. One is how we're gonna leverage the representativeness of the real data to help generalize the RCT findings to broader and richer patient population. On the other hand, we can also go an opposite direction. That means we can borrow information from real data to improve the RCT analysis efficiency, right? So this is going a different direction. But here, we need to be very careful. We don't want to borrow biases, right? So we only want to borrow useful information from the real data. So the key here, the key issues we want to address is to avoid biases in the borrowing procedure. And we have been developing different strategies. For example, the control variate approach, test and pool bias modeling, selective borrowing approaches that can recognize the biases in real world settings. And then we want to borrow the useful information to improve the RCT analysis. So this is some statistical toolkit we have been developing. And then in the rest of the talk, I'm gonna introduce hybrid control designs as an example where we can borrow real world external controls into the RCT. And we're gonna introduce a bias-aware frequentist toolkit to illustrate the framework of causal inference. So here, before I talk about hybrid control designs, let's recall what is the advantage of RCT. So for the randomized clinical trial, for a group of patients, we actually have a randomization of the treatment. This ensures different treatment groups are comparable, right? So we can compare apples to apples. And because of the randomization of the treatment, we can directly compare their outcomes. If we see any differences between the two treatment groups, that is due to the treatment effect instead of other variables. So this is called the internal validity. We can also utilize the covariance information. For example, we can use ANCOVA as a covariance adjustment to boost the efficiency of the naive comparison. And we don't rely on the ANCOVA model to be correct, right? This is because of the randomization of the treatment in the design stage. This is called the model robustness. But now if we replace some internal controls with external controls, they can come from the real world settings, it breaks randomization, and therefore we may not have the internal validity or model robustness anymore. And here we only replace part of the controls with external controls. This is because full substitution is risky, right? Because if we do full substitution, we don't have any benchmark to compare whether like the real controls are similar or not. And hybrid control designs could be more effective involving small RCT controls. And this approach combines randomization from RCTs with additional controls from real-world data. And this helps evaluate the potential biases in real-world controls and facilitate the information borrowing. So the next question becomes how we can borrow comparable external controls and discard or downweight the incomparable external controls. This actually is not a new question. Bayesian analysis actually has been developed for such questions a decade ago. For the Bayesian methods, it tried to downweight, they use external controls when there's like discrepancy arises from external controls and the RCT controls. And this can be achieved by different power priors, like prior designs, for example, power priors, commensurate priors, so on and so forth. But for the Bayesian methods, it is not clear about the statistical operating characteristic, for example, type 1 errors, so on and so forth. This motivates us to develop like some frequentist methods. So here I'm gonna introduce the bias-aware borrowing approaches frequentist toolkit. And we have different strategies to control biases, for example, test and pool, bias function modeling, selective borrowing with the semi-parametric frameworks or randomization-based inference framework. So in the rest of the talk, I'm gonna briefly introduce the topics here. And then I will give some high levels of the different strategies. But if you are interested in the topics, I will provide some references in the end. So test and pull, actually, this idea is quite intuitive because we wonder whether the external controls and the RCT controls are comparable or not. We first formulate a hypothesis, right? So here in the first step, we test for the differences in outcome between the RCT controls and the external controls. And the hypothesis is that the outcome means are actually the same in the concurrent controls, what we call internal controls, and the external control. And then the alternative says they are different, right? So if the null hypothesis is not rejected, that means there's no sufficient evidence in the data suggested that the external controls have bias. Then we want to pool the RCT and external controls because we can borrow some useful information from the external controls. So we can conduct the pooled analysis. On the other hand, if the null hypothesis is rejected, so that means the data suggested that there is sufficient significant biases in the external controls. So in this case, it's safer to just retain the RCT controls for the analysis, right? We don't want to borrow biases from the external controls. So the idea is quite simple and very intuitive. Actually, this is the most common approach in statistics in common practice. For the test and the estimation, we can actually use doubly robust causal inference methods that can utilize some information or some machine learning techniques. So this can be very flexible. But there are some limitations associated with the test and pull strategy. We actually find that we have double dipping problem here, right? Because we use the same data for testing and also estimation. So this brings in the post selection problem here. And we show that in order to correct this, sometimes we can be very conservative in inference procedure. And the test and pull require the strong signals of the violation and the moderate sample sizes or RCT controls in order to perform well. And sometimes we reject the whole external controls. This can be wasteful, right? Because even like the whole studies have the biases, some of the external control units may still be comparable, right? So whether we can still utilize the biased external controls in this case. This leads to the second question we want to address. Can the data from biased external controls still useful to improve the RCT analysis? And we are motivated to address this question. We come up with the two solutions. The first one is a bias function modeling. So we create a mathematical bias function to model and subtract the bias from the external control, and therefore we can correct the biases. And in this case, possibly we can still borrow some information from external data. And the second one is selective borrowing. So imagine that like some external control units are still useful. We want to borrow such a unit, right? Although that the whole studies may have a bias on average. So I will introduce the two approaches here. So the first is bias function modeling. Let me use the ANCOA model as an example. So in the RCT, for the RCT data, we can safely invoke the ANCOA model. This is just a working model. Because of randomization, we don't necessarily assume the ANCOA model is a correct model, right? But now, because we have external controls, this breaks randomization, and therefore simple ANCOA model for the joint data, so RCT combined external control, may not be valid anymore, right? So it can produce biases because we don't have randomization anymore. So what is the solution? The solution actually is very simple. Because the ANCOA model applied to the external control is not valid anymore, we want to adjust this bias. So we add a bias adjustment to the ANCOA model. So this bias function is defined as the difference between the external control outcome mean and the working ANCOA model. This can quantify the differences between the external control and the working model in the RCT. Because of this bias correction, then the model, so the joint model, so the ANCOA model augmented by the bias function is a correct model for the external control. Then this entails a joint model for the RCT plus external control. So this leads to the joint modeling in RCT and the external control, the combined data. We call this ANCOA augmented by the bias function. So you can see for the RCT, we still have the ANCOA model, and S is the study indicator. So S equals 1 means the RCT, S equals 0 means external control. Then adding this term means that we augmented the ANCOA model by the bias function for the external controls. And this can capture the true outcome mean in external control. Because the bias function is unknown, so we approximate this by some spline functions or some other basis expansions. So we require this part to be sufficient enough so that we can approximate the bias function well. But you can see this is the joint modeling for the combined data, and we can use the least square estimation to provide the consistent estimator for the average to mean fat. So from the joint model, we just use the least square formulation plus some regularization parameters. And the first regularization parameters is for the covariate adjustment in the RCT. For example, you have a high dimension of covariates and you want to select the important variables. We can use the penalty, for example, lasso penalty or other penalties to select the important variables. The second term actually is more important because it regularizes the bias function. So we can encourage the smoothness or simple structure of the bias function so that we can borrow some information from the external controls. This is because we want to have the external control information to identify the bias function. And at the same time, we want to borrow like some remaining information from external control to improve the efficiency of the ANCOVA analysis. Because the treatment coefficient actually it dictates the average treatment effect here. And therefore, if there is like additional information in the external control, it can boost the efficiency of the beta A estimation. So here you can see we don't actually rely on any model assumptions, so assumption lean. And also the bias function captures all possible external control biases, for example, due to unmeasured confounding, due to outcome invalidity, so on and so forth. So all the bias sources we mentioned previously. Also, it can always gain efficiency compared to the RCT only ANCOVA analysis. So this is a very promising result. And if the bias function is simple, so for example, you can think about the bias function is exactly 0, we can gain a lot of information from the external controls, because in that case, the external control information actually contributes a lot to the beta A estimation. So this is very good news, but the limitation of this framework is that we just invoke the ANCOVA model because this is convenient in practice, but the statistical analysis methods may not be optimal, right? So if we go after statistical optimality, what kind of options we can have? At the same time, we also want to adjust the bias from the external control borrowing. So here comes the semi-parametric efficiency estimator with the selective borrowing procedure we have been developing. So we want to study the statistical optimality, and therefore it is very helpful to first list all the assumptions we required. And here we have assumption 1 and 2. So assumption 1 is about the RCT validity. So it basically tells that we have randomization of treatment and the positivity of the treatment assignment. This is usually held by trial design, and therefore we assume assumption 1 hold by default. Assumption 2 is about the whether we can borrow external control or not. And we call this external control outcome compatibility, in the sense that the outcome mean function in the external control and the outcome mean function in the RCT control is comparable, is exchangeable. So that we can borrow some like outcome relationship from the external control to improve the efficiency of the RCT analysis. But you can see from this assumption, we don't impose any conditions for or restrictions for the covariate distribution, right? So we allow possible covariate shift between the external control and the RCT. But we don't allow for other sources of bias, for example, unmeasured confounding biases, measurement errors, or the invalidity of the outcome measures, so on and so forth. So if any of these sources of bias occurs, this assumption is violated. So you can see this assumption actually is a very restrictive, and we view this as an idealistic assumption. And this leads to two objectives of our framework. So in the first step, we are willing to assume this assumption hold. So the external control outcomes are actually comparable. And under this assumption 1 and 2, we want to develop efficient and robust estimators for the average treatment effect in the RCT patient population. And then in the next step, we consider this assumption may be violated. And this is very realistic scenarios. In this case, we want to develop some selective borrowing approaches so that we can borrow on the comparable external controls, so that we don't introduce biases in the first step, right? So once we have selected these comparable external controls, we can apply the step 1 estimator for the comparable external controls to mitigate biases because of the outcome drift. So if assumption 2 is violated, we assume that there is an outcome drift problem. So let's go to step one first, we want to adjust the covariance shift, and we are operating under the case where assumptions 1 and 2 hold. And we developed this estimator called augmented calibration weighting estimator. This is actually motivated from the semi-parametric efficiency theory. The equation looks a little bit ugly. I don't want to go to the technical details here, but I will mention a few parts. So this part, the Q part, actually is adjusting for the covariance shift between the external controls and the RCT controls. And R here is the variance ratio, the variance ratio of the outcome given covariance between the RCT controls and the external controls. So this motivation is that because we assume the outcome mean functions are the same between external controls and RCT controls, but we also need to weight the external control differently if we see large variability in their outcomes, right? So if the outcome measure has a lot of noisy, we also want to downweight the use of the external controls. So this component comes naturally from the semi-parametric framework, and this makes a lot of sense to have such a component. But using the semi-parametric theory to motivate such estimators has a clear advantage because we can show that such estimator had the double robustness, local efficiency, and the flexibility to adopt nuisance, like machine learning or AI for nuisance parameters. So for example, you can see when we construct such estimators, we need to approximate a lot of nuisance parameters, right? So for example, outcome mean function here, the weighting function here, so on and so forth. And AI and the ML actually has this advantage, so we can approximate the nuisance parameters well using AI and ML. But at the same time, we want to get clear statistical properties about the average Tremaine effect estimator. And the doubly robust estimator has this advantage. So even we incorporate machine learning or AI for nuisance parameters, we can still get the routine consistency or symptotic normality for the tau hat, so average Tremaine effect estimation. So this really shows the causal inference methods has this advantage to incorporate very powerful machine learning approaches. So that's the step one. But step one is derived under the assumption that the outcome from the external controls are comparable. What if this assumption is validated? So we have the outcome drift problem. If we naively apply the step one estimator without addressing this outcome drift problem, we get the worst case estimator, right? Because the estimator is very accurate, but sorry, it has a very small variance, but if it is biased, then it concentrates at the wrong place. So we want to address the outcome drift so that we can avoid biases from borrowing. And the idea here is very simple. In step two, we want to do selective borrowing. So if we see biases in some external control unit, we don't want to borrow such unit. We only want to borrow the external control unit with small bias. So to do that, we first assign a bias parameter for each unit in external control, BJ here. So BJ actually measures the difference in the outcome means between the external control unit J and the RCT control unit with the same covariance XJ. So if this BJ is 0, that means this external control unit actually is exchangeable with some RCT control unit. Otherwise, this external control unit has biases. And then we only want to select such external control unit with bias approximately 0. And then once we have this external control set, we can apply the step one estimator for this RCT plus the selected external control set so that we can prevent the hidden biases, right, from, for example, unmeasured confounding, so on and so forth. Now the question is how we're going to select out such a comparable external control unit. We can actually translate the selection into penalization. This is because for the bias parameter, we can actually construct some initial estimate. note that the bias parameter actually is identifiable because we have the RCT control, the concurrent controls. We have benchmarks, right? So we can first calculate the bias parameter from some initial estimates. Okay, and then some of the initial estimates are small, some of the initial estimates are large, and then we want to use the penalization technique to shrink the final estimate of BJ toward zero if some of the initial estimates are small, right? And then once we do this, some of the final estimate of the bias parameter is zero. So these are the external control units we want to select. And for the penalization, this actually is very common in statistics. We can use adaptive lasso, SCAD, and other penalties. So this is the typical variable selection technique, but we translate the selection of external controls into the penalization so that we can utilize a lot of off-the-shelf statistical methods for that. So once we selected this comparable external control set, we constructed the ACW estimator in step 1, and we call this adaptive ACW estimator. Here, I provided some plasmode simulation. The goal here is to assess the RCT only estimator because we want to compare to the RCT only analysis and see whether we can gain efficiency or not. And then the naive ACW estimator, so that means we naively combine RCT and external control and apply the optimal estimators. So you can imagine that this estimator will be the best if there's no biases in external control. And then the adaptive ACW estimator, and that means we first do selection based on the selective borrowing procedure I just described, and then apply the ACW estimator. Then we'll compare. We want to assess the 3 estimators in terms of type 1 error control, the power in various realistic settings. And here we have the simulation designs, and this is motivated from actual oncology RCTs. I want to mention that we generated the outcome in 2 scenarios. One is assumption 2 holds, and one is assumption is violated due to unmeasured confounding. This scenario actually is more realistic in practice. And then we vary the sample sizes. So we vary the RCT controls from 20 to 100. RCT treatment remains at 200, and external control size remains to be 3,000. And here the result for the first case where assumption 2 is not violated, in the sense that the outcome functions from external control and RCT controls are exchangeable. And we have different panels. We have bias, variance, mean square error, type 1 error, and also the power for a different estimation. We have AIPW estimator that is RCT only analysis, ACW estimator, and adaptive ACW estimator in different colors. And now if we examine the bias panel and the variance panel, you can see ACW estimator actually is the best, right? It has the smallest bias and the variance, and it gains the efficiency over the RCT only estimator. It can also control type 1 error well, achieve high power. But adaptive ACW estimator is also good, right? It can have small bias and variance, gains the efficiency over the RCT only estimator. It also controls type 1 error well, achieves a high power. But the advantage of the adaptive selection really shows here in the second scenario where assumption 2 does not hold due to unmeasured confounding. Now you can see ACW estimator become the worst. It has the largest bias and the small variance. So that means it's the worst case scenario, right? It cannot control type 1 error. It has low power. But adaptive ACW is still good because of the selective selection procedure we have. It can control the bias well, so it can also have small variance, gains efficiency over RCT only, and it controls type 1 error well when at least like the NC size, NC is the concurrent control size, is relatively large, and it can achieve good power. But one limitation is that the type 1 error control is still very poor for small samples, right? So you can see here for adaptive ACW estimator, it cannot strictly control type 1 error when NC size is small. So this motivate the question here. So can we still control type 1 errors and maintain power gain in small samples? To achieve this, we utilize the Fisher randomization test. This is a test that guarantees valid result by comparing the actual result to thousands of random reshuffles of the treatment assignment, because the treatment assignment mechanism actually is known in RCTs, right? And in this procedure, randomness only comes from randomization of the treatment assignment, and all others are considered as fixed. So we don't need to make any model assumptions. And therefore, Fisher randomization test actually is a very good procedure. And here is a general procedure for the Fisher randomization test. We actually need to change the question a little bit by assuming the sharp null hypothesis. Basically, assume that there's no individual treatment effect in RCT. And then we want to test whether this null hypothesis is true or not for the treatment. Okay, and then in order to test this hypothesis, we need to have a test statistic. And Fisher randomization test actually is very flexible, so you can invoke any test statistic that can constructed from the data. And then here we use the average treatment effect estimator. Okay, and then we need to construct some reference distribution so that we can gauge whether the realized value of the test statistic is extreme or not, right? And then the reference distribution actually comes from the randomization of the treatment assignment mechanism. So this is where the randomness comes from. But the treatment assignment mechanism actually is known in RCT because we can control the trial design. So we know what is the randomness mechanism. So we can permute all the treatment assignment mechanism from the actual trials to get the randomness of the test statistic that forms the reference distribution, and therefore we can calculate the exact p-value. And in the end, we can use the exact p-value to see whether the test statistic, the realized value is extreme or not under the null hypothesis, right? And therefore we can reject or accept the null hypothesis. And the validity of the Fisher randomization test is established here. We can control type 1 error rate for testing the sharp null hypothesis regardless of the sample sizes. So we don't rely on a sample size to go to infinity. It is a finite sample exact. And also we don't make any distribution assumptions. So the Fisher randomization test is distribution free and therefore robust. And it holds for any test statistic as long as the permuted treatment assignment aligns with the true assignment mechanism. And the good news is that FDA is open to using permutation-based tests, providing a clear path for real-world applications in this scenario, and we can achieve a strict type 1 error control. And for Fisher randomization test in our context for data integration, in order to guide us how to utilize the additional information from the external data sources, we conducted the power analysis. And what we find is that if the test statistic is consistent, then a smaller variance of the test statistic can lead to higher power. So therefore, we can chase a consistent and efficient test statistic in this data integration context. So we can consider no borrowing, so that's the RCT only test statistic, or full borrowing. For example, we can use the ACW estimator combining the RCT and the external controls, or the selective borrowing procedure. So that's the ACW estimator with a selective borrowing procedure. But here, differently from the previous result, we use conformal inference to select because conformal inference is also distribution-free selection procedure. I will omit the selection details, but if you are interested in how we're going to select the external control unit in a robust manner, you can check out the paper here. The catch is here. So we know that the Fisher randomization test remains valid, even though we borrow some biases from external control, um, right? Because Fisher randomization has this validity guarantee. But what is the consequence of borrowing bias external controls in the Fisher randomization test? So here, in order to illustrate this, we generate the case where outcome indeed have incompatibility or hidden biases in external control. And we calculate the Fisher randomization test for a no borrowing approach, full borrowing approach, and a conformal selective borrowing approach in the figures here. And here, these figures shows the exact p-value distribution. You can see across different test statistic, even we borrow some biases from the external controls, the p-value distribution actually is very uniform. And you can see different statistic can also control type 1 error very well, even though for the full borrowing, we actually borrow the biased external controls, right? And the consequence actually shows in the power calculation. So here under the alternative hypothesis, you can see borrowing incomparable external controls leads to severe power loss compared to the no borrowing approach. And if we borrow only comparable external controls, we can actually restore the power gain. So that is the advantage of using the conformal selection, select only comparable external controls in constructing our test statistic. So you can see if we use the ACW estimator with conformal selection as our test statistic, we can control the type 1 error strictly, and also we can restore the power, and then we can gain power compared to the RCT only analysis, the no borrowing approach. And we indeed conducted some simulations to assess the finite sample performance of the Fisher randomization test. I won't go to the details, but point out the two figures here. One is type 1 error and one is a power for different scenarios. So the x-axis illustrated the magnitude of hidden biases in external controls. So again, we have no borrowing approach, a full borrowing approach, and a conformal selective borrowing approach. So you can see across the scenarios, we can control type 1 error very well, right? So this is the benchmark. This is a 0.05 level. On the test statistic can give very good type 1 error control. But very interestingly, for the power analysis, you can see compared to the no borrowing approach, the full borrowing approach, as the bias increases, actually reduce the power a lot, right? It's actually monotonely decreasing as the power increase, as the bias increases. However, with our selective borrowing approach, you can see it first decreases and then restores as the bias increases. This is the challenging case in the middle. So you can see like in some challenging cases, we can still lose some power compared to the no borrowing approach. But this is anticipated because in those cases, the bias magnitude is not that large and not that small. And therefore, it is very hard to determine whether we need to borrow such an external control unit or not. So this introduces uncertainty, and therefore, in those cases, we lose some power compared to the no borrowing approach. But when the bias increases, we can actually detect such strong signals and restore our power. So this is advantage of our conformal selective borrowing. And this phenomenon actually is not new in the statistical literature. It has been in other like selection procedures as well. And now let's summarize the bias-aware borrowing approaches that recognize the benefit of RCTs and RWD, as well as the strengths of statistical thinking and AI/ML. So we employ the methods like the pre-testing, bias function modeling, or selective borrowing to reduce the potential biases from borrowing. And our test statistic or estimators are designed to be doubly robust so that we can leverage AI and ML predictive power while remaining consistent and have good statistical power, statistical properties as well. So I apologize for not having time to detail everything here, but if you are interested in more information, I will provide references in the end. And looking forward, there are many challenges, which also means opportunities. So, for example, data privacy and confidentiality, uncertainty quantification, small sample problems, also data quality issues. And looking forward, I see numerous potentials in federated learning, privacy preserving analytics, and also adaptive evidence integration. And these approaches can help us move forward with a more personalized, faster, and safer drug development. With that, I'm gonna stop here, and I welcome any questions. Here are the references.
That's great. Thank you very much, Shu, for a fascinating talk. There's already a couple of questions in the Q&A, which we'll go through, and I would like to encourage other attendees to keep posting their questions in the Q&A here, or alternatively the chat, but if possible in the Q&A, because that's easier for me to keep track, but I'll try to watch the chat as well as the Q&A. We have a question here from Peter Missenbrink, and apologies if I'm mispronouncing your name. And he's asking, how large does NC need to be for the adaptive ACW to perform reasonably well?
Yeah, this is a very good question. We actually have some simulation studies. Basically, it requires that the NC size to be relatively large so that we can do a good job in selection. So you can see from this scenario, this is, of course, like under our study design here, how we generate the data. But you can see when NC is around like 60, it can control type 1 error relatively well. But it really depends on like whether the data is complex or not, right? So this result may not be generalized to other cases. But the overall conclusion is we still need a relatively large concurrent controls in order to do a good job in selection.
Mm-hmm. And there's also a follow-up from Peter as well, and he's mentioning the Fisher randomization test. And he's saying that you state that there, everything is good as long as AB aligns with the true randomization A. But how is this alignment defined for the assumption to hold?
Yeah. So this is actually not an assumption because in RCT, the treatment assignment mechanism actually is known to us. So we only need to permute AB according to the actual design.
Mm-hmm.
So if you are the experimenter, you know the design, we can just permute AB accordingly, and therefore it can generate the reference distribution for the test statistic.
Mm-hmm. Perfect. Well, I guess I have a bit of a more general question before I go over to the Q&A box again, because you're mentioning integrative analysis, and then you showed us a few of the pros and cons or the basic descriptions of control trials and then external data. But I was just wondering, is there any sort of quality control that you do before you incorporate any type of external data into your results?
Yeah, this is a very good question. I think FDA also produces some guidance about how to use the external controls because you can imagine that if the external control has a lot of biases, you don't want to utilize them, right? So according to FDA guidance, one should definitely assess whether the external controls are good quality or not, whether it is fit for purpose or not. So one can check whether the external control are relevant for the study question or not. So for example, whether the treatment outcome or covariate definitions aligns well with the trial definitions, right? And also whether the data are reliable or not, because in a lot of cases, there may be a lot of missing values, right? And in measurement errors, or like the study period differ a lot between the external controls and the RCT studies, then all these can raise like concerns with utilizing the external controls. So I think it would be a best practice like to check whether the studies are relevant or reliable before naively utilize applying any methods like for data integration.
And this is especially important in a world where we have big data and big data methods and machine learning methods that would definitely reproduce what we see in the data. So if the data is not good, then your estimator is not going to be good, right? So, and I guess, um, as I mentioned, machine learning, we see those techniques gaining a lot of popularity now. So I was just wondering if you've done any work where you, or you plan on doing any work where you use some sort of black box algorithm to estimate the important quantities you're looking at?
Yeah, this is a very good question. So AI ML is playing an increasingly important role, right, in the drug discovery context, especially if we want to operate under the real world evidence and real world data framework, because real world data requires some new techniques because it has complexity, high dimensionality, high volume, so on and so forth. So I think the AI ML can do a very good job in capturing complex relationship. This is a good fit, right, to address the challenges from the real world data and the real world evidence. But also, it has some limitations, as you mentioned, that this is like very complex model. It is black box, and it does not have clear interpretability. But
Mhm.
this is where the principle, the statistical methods or causal inference methods can come in, like the double robust estimator I mentioned. This can utilize the AI or ML to approximate the nuisance parameters well. And at the same time, we can show that the ATE estimator, so the estimator for the average treatment effect, still can have very clear statistical properties. And
Mhm.
therefore, I think this is how statistics plays a crucial, important role in AI and ML research. We need to combine the conventional wisdom and the new techniques here.
Yeah, I completely agree. And speaking about robustness, there's a question here from an attendee, but it's anonymous here. But if you'd like to identify yourself in the chat, feel free. Otherwise, it's no problem. But they're asking if these methods are robust to how the external data source is selected. So, and they give an example. So imagine there's a sponsor and they know the outcomes of the external data source before they select that data source. So how robust it is to this types of bias. I imagine that's what this person's trying to ask.
Yeah. So this is a very good question. First, as I suggest, like if we want to utilize the external controls, first we need to have some pre assessment, right, about whether the data are relevant, whether the data are reliable. After that, there may be still issues like biases hidden in the external controls. Or for example, there may be unmeasured confounding issues you cannot check based on the data, then our selective borrowing approach actually is exact doing this. So we are filtering like the biases due to hidden biases, for example, unmeasured confounding. So even you see some outcome from the external controls, this selective borrowing is still very important, like to mitigate the like biases due to hidden sources.
Mm-hmm. And following up on the bias issue, so we have a question here from Taiwa Choi, and apologies if I mispronounced your name, but they're asking if RCT and EC were collected in different time periods. So for instance, there could be some calendar bias. Do you have any remedies that can handle this type of situation?
Yeah, exactly. So the bias-aware approaches are exactly filtering out such biases as well. So for example, if there's a calendar bias, the outcome mean function from the external controls and the concurrent controls will be different. And therefore, our selective borrowing approach, for example, can select out such external control unit. So this is like a selective borrowing approach. Bias function modeling can also like model what is the difference between the external controls and the concurrent controls due to the time shift. And utilize this bias function, we can correct the bias from external controls. And hopefully there is like still some relevant information in external control can boost the efficiency for the treatment effect estimation.
Perfect. I mean, I do have a question that I was thinking. I would like to get your opinion on this and your view on this because there are many early career statisticians and biometricians watching us right now and will watch us in the future from the recorded session. And as part of our Distinguished Lecture Series, one of our objectives is to inspire these early career statisticians and biometricians to follow their career in biometry. And one thing that we have seen in open forums, in actually very high impact journals right now, is the issue about p-values. So we see groups of statisticians and also applied scientists completely saying that p-values cannot be used, should not be used at all whatsoever. And there's other groups saying they should, but with the caveats that come with p-values. So for instance, there's a recent, I think in 2018, we had the paper Redefine Statistical Significance, or a group of statisticians were talking about using 0.005 instead of 0.05, but then of course coming with all the caveats. But then I think there was a paper by Sandra Greenland, who was actually one of the keynote speakers in the last IBC, where they say remove rather than redefine statistical significance, again, with all the caveats that we know about. But I would just like to know your perspectives on p-values and the magical 0.05 for those who are early career statisticians and biometricians here looking for a voice to follow.
Yeah, I think this is a very good question. And I think this topic received a lot of debate among statistics and also other related areas. I think right now for this context data integration about the treatment effect evaluation, I still advocate using p-value. This is because we don't have like other challenging issues to handle, for example, multiple testing problems. Then I think the traditional p-value can still play an important role here to decide whether the treatment is effective or not. And if there are like other challenging cases, for example, we are testing a lot of hypotheses, then it generates concerns that utilize the p-value without any adjustment. Also, type 1 error controls from the FDA perspective is still very important. And this is related to like p-value or significance levels and so forth. But I think some statistic traditions still is important and may still like play an important role down the road.
Yeah, absolutely. Thank you. We have another question here on the chat by Jack Lee. Could you comment on striking a balance between the generalizability and the efficiency? And so they explain the stronger assumption is the more efficiency gain, the more efficient it will be, but less generalizable and less robust. Yeah, could you comment on that?
Yeah, this is a very good question. I think this is a limitation of the RCT because usually RCT involve very restrictive enrollment criteria so that it can enrich the patient population and therefore gain more power. But also it limits the generalizability so that the RCT findings cannot be generalized well to like broader and richer patient population. How to balance this? I think currently, for example, programmatic trials that targets like generalizability of the RCTs and also like recruit or enroll patients in a sufficient rich manner. And then we can also use enrichment designs so that we don't lose like a lot of power for such designs. Yeah, I think this is a very good question, and it actually is an ongoing discussion in the community as well.
Great, thank you. There's a person here asking about the presentation document. So I was just wondering, Shu, do you have any personal web pages where you have your slides available, or are they available anywhere?
For the slides, I haven't made this public, but I can provide other slides if.
And at the same time, this will be recorded for members as well in the members area, as Peter mentioned before. So if you're an IBS member, you can have a look at the recorded presentation. There's a question here from Hui Yi, and they say, I'm new to the area of RCT and RCT and RWD, and could you please share a little bit more about the requirement and formality of the RWD, as in how do we assess the informativeness and biasness of RWD either before or after the integration?
Oh, this is a very good question. I mentioned that there can be a variety of sources for RWD. For example, patient disease registries, EHR system claims, or even some national health studies. They can provide population level of data. This data sometimes can be publicly available, sometimes is restrictive. But I encourage like if some sponsors have this data, it is better like to share them with the researchers so that it can be utilized later on for treatment evaluation more quickly. And then how to assess the quality of the real world data. Firstly, we have all these causal assumptions required. These are actually the guiding principles to choose the right data to study for data integration. So for example, we require the exchangeability of the outcomes. Then one need to assess whether the outcome definitions are similar between the two studies or not, right? Not just the outcome, but also the other variables. Also the like the previous participant asked about the time issues, whether the times are sufficiently close or not, whether the care, the standard of care changes or not, like between different time periods. So all these aspects should be checked in order for the data integration method to be successful. I think the FDA provided a very good guidance about how to assess the fit for purpose external control or the real world data in order for the cost inference method to work. And these criteria also applies for this case when we do data integration.
That's great. Thank you. I do have another question. If you have a question, please put it in the Q&A. We do have a few more minutes to discuss with Professor Xu. So I was just wondering, Xu, in terms of censoring of observations, if that's easily incorporated in the approach, or would you have to maybe create some sort of joint likelihood approach, or I'm not sure. Have you worked with some censored observations? Because I mean, this is in clinical trials, you have a lot of censoring sometimes.
Yeah. This is a very good question because time to event data, I think it's the most popular ones from the clinical trials. And there can be like race sensor data or other sensor type of the data. We indeed have some approaches that can be extended for the survival data. So for example, so here we have a recent paper published in Biometrics. So this is about bias function modeling for race sensor data when we synthesize randomized clinical trials and real world studies. Here the focus is about heterogeneity of treatment effect, but the outcome is a race sensor data. So our approach, although I used the continuous outcome or non-survival outcome as an illustration, but some of the methods or the key ideas can be transferable to like a race sensor data as well.
Perfect. And I do have another question, if you allow me, because we see a lot of studies that utilize meta-analyses of separate randomized controlled trials. But I was just wondering, I would assume that that's true. That the case would be that if you incorporate other data sources within a meta-analytic approach, you could probably obtain even more robust results. Of course, given that all quality control is done beforehand. So have you had any experience with the meta-analytic approaches and merging with the data fusion techniques you were mentioning?
Yeah, I can imagine like after two bias corrections for like different data sources, and if the bias corrected estimators follows the meta-analysis assumptions, we can still like do like some borrowing or integration. But I think that the major part of this data integration context is like to remove bias from the external data sources so that they can target at the same estimate of interest. And then we can like do fusion or like other techniques to still like boost the efficiency.
Perfect. Thank you. I don't see any other questions in the Q&A, so I would. Oh, there's another one. Sorry. As I said that, a question popped up. And I do have a final one after that. So this is a follow-up on an earlier question about the assumption and outcome. How can you avoid a self-fulfilled prophecy? That's a nice one. For instance, you only select the RWD that's similar to the current finding and reinforces the status quo. Or could you say a little bit about broadening the scope, such as including more trials in a network type meta-analysis?
Oh, this is a very good question. I think this is a different, let's see, different topic. In the sense that current methods I'm talking about try to borrow information from RWD to boost the efficiency of the RCT analysis. So we've restricted the target estimate to RCT patient population. But if we will want to, sorry. If we want to generalize real-world evidence, for example, make the RCT findings more generalizable to other like patient population, whether the patient population is represented by real-world data or the trial networks, we can go an opposite direction. So here, the methods we're developing try to broaden or extend the RCT findings to other patient population. So I think these are the two different questions. How to combine them, I haven't thought about this, but this definitely is an interesting topic.
Great. Well, I do have a final question then for you, more like an overall general question. What would be the message you would have for the early biometricians, early career biometricians and statisticians who are very interested in working in biometry in terms of health and control trials and so on, and but perhaps they feel a bit it feels daunting to them for the plethora of material that's already available and all the techniques that already exist. And what's your message to those who are beginning in this journey?
Yeah, this is a very good question. I think during my early career, my habit is like sitting in my office and think about the statistical problems. And one time I'm working on very interesting topic on my own, and it's very hard for me to find a real world data like, to illustrate. I thought, oh, this is like a very common scenarios, but surprisingly, it's very hard for me to find the like real data for illustration. And that motivated me like to first to find the problems of interest that can be practically very important. And then we can think about like how to develop a method to solve like a statistical challenges there. And in order to do that, I actually have a lot of collaborators from pharmaceutical companies so that I can initiate some discussions with them and to understand what are the real problems they encounter in their practice. And then we can form a team, talk about the real problem and what are the challenges in the real data. And then I can contribute thinking about how to develop a statistical method to solve those problems. So I think the first step is to initiate some discussion with practitioners and then identify the important problem to work on. Don't just sit in the office and scratch our heads and think about like possibly realistic problems, but sometimes it will be very surprising that you cannot find the real data to illustrate the statistical methods.
That's a great advice. Thank you very much, Shu. This was a very successful event. I see at one point we had almost 100 attendees and a very engaged audience. So I thank you all for this. And I'd like to pass it over to Peter.
Okay, very good. Thank you. Well, thank you to both of you, actually. And I'd like to thank all of our audience members for joining us today. We will be happy to answer any additional questions through the IBS members community. It's always there, 24 hours a day. And please feel free to post any remaining questions submitted that we weren't able to answer using that feature. A recording of this session will be made available hopefully next week under the education tab. Just look for the recordings area in the main navigation of our website. And just remember that you will need to log into your member account to access this and other video sessions. As many of you know, We're already in the midst of a busy conference season with many local events coming up. We encourage you to visit the IBS events calendar, which has just recently been refreshed as part of the launch of our new site, for any upcoming opportunities, and we are adding additional events daily. We hope to see many of you at our next Journal Club session. That'll be our next virtual session, and that's taking place on the 17th of October, addressing Learn As You Go trials. That is my new favorite phrase, actually. Optimizing treatments and preventing trial failure through ongoing learning. Registration is complimentary to members, and so you can sign up today to reserve your spot. We are at the end of this session. So again, thank you to all for joining us, and you may now exit. We will see you next time, hopefully at another distinguished lecture or journal club or something else. Thanks very much.
Thank you, Peter. Thank you, Rafi.
Thank
Judith
you.
actually is my postdoc advisor.
Oh, really? Oh, that's excellent. Okay, so
Yeah.
you'll be in the audience, right, for that day? Very good. Okay, we'll see you all then. Take care.
Take care. Bye.
Bye.